And then I'm going to give some instructions before we get started and before we are streaming this live to Facebook. This is our first AD seminar series that we're doing as a webinar. And with that, the way that we have questions answered will be a little bit different than our last AD seminar series at the Mesulam Center. So you can either input your questions in the Q&A box that you'll see on your toolbar. And then we do also have the ability to allow people to talk. So if you would prefer to ask your question, we can let you in and we'll be able to see you and you could verbally ask your question. So just send a Q&A to the Q&A bar or the chat feature if you see that when we get to the Q&A part of the seminar series, which will be the last 15 or so minutes. And with that, I would like to introduce Dr. John Disterhoff and Dr. Robert Vassar.
Okay. So it's my pleasure to introduce Dr. Robert Vassar, Bob Vassar, this morning. Dr. Vassar is the Davey Professor of Alzheimer's Research in the Department of Neurology at Northwestern. He's the Scientific Director of Behavioral Neurology, also in the Department of Neurology, and the Director of the Alzheimer's Disease Core Center, the P30, that's within the Mesulam Center, but also, of course, at Feinberg School of Medicine. I thought I would give just a very brief introduction. I think probably most people participating this morning know Bob and know of his work, so Bob received his PhD in molecular genetics and cell biology at the University of Chicago. He then did a postdoctoral fellowship with Nobel laureate, he wasn't a Nobel laureate at that point, but Richard Axel at Columbia University. Joined Amgen after his postdoc, and then following his stint at Amgen in Southern California, came to Northwestern, and he's been with us since. So Bob, a couple of highlights of his research career. He's been looking at or investigating the cell biology, molecular genetics, and biochemistry of AD for more than 20 years. I think he's best known or well known for the fact that he co-discovered and characterized the beta secretase enzyme BASE1, and it's a prime therapeutic target for AD, and there are several compounds that are in phase 3 trials right now that are focused on BASE1. He's also generated in his lab conditional knockout mice to validate BASE1. And one thing that I keep seeing, I mean, that is noticeable and I know about was the fact that he generated a transgenic mouse line called the 5XFAD mouse, which is widely used in AD research, both in the United States and throughout the world. So Bob has received several impressive awards. Let me just mention a few of them. The MetLife Foundation Award for Medical Research in Alzheimer's Disease, the Potemkin Prize for Research in PICS, Alzheimer's and Related Diseases. He's a AAA American Association for the Advancement of Science Fellow. He's involved in the Dana Alliance for Brain Initiatives, and he was a Zenith Fellow. He received that award from the Alzheimer's Association. Notably, in these days of tight research funding, Bob's lab is supported by 3 separate R01 type grants that he is the PI of. He's also, of course, the director of the Alzheimer's Disease Research Center grant, the P30, and he has several foundation awards that is supporting research in his very active laboratory. One final thing I should mention is that Bob is the co-director of the Mechanisms of Aging and Dementia T32 training program. So with that, let me turn it over to Bob, who's going to tell us today about insights into the role of angiotensin converting enzyme in Alzheimer's disease. Bob, take it away.
Well, thank you, John, for that kind introduction. Let me share my screen. Oh, it says that the host disabled the attendee screen sharing. So, Lisa, could you fix that for me?
Yes, I just fixed it.
All right, great, thanks. So let me share the screen. So what I want to tell you today is a story that was recently published in Science Translational Medicine. And okay, now I'm having, Lisa, that same problem that we were worried about just happened. Let me stop my screen sharing. I was having some technical difficulties the other day with my PowerPoint. Let's see if I can.
Do you have your mouse on the second monitor?
Now it's working. Okay, great. Can you see my screen? Is it moving?
You just have to share it.
No, I haven't. Okay, hold on. Sorry for the technical difficulties here. All right, let me try this again. All right, let's see if this works. Great, now it's moving. All right, great. So what I want to give you, I'll tell you about this story that was recently published in Science Translational Medicine. I first want to give you a little bit of background about angiotensin converting enzyme and its links to Alzheimer's disease. Then I'll go into the actual story of the results of the study that we did on this rare coding variant in angiotensin converting enzyme, arginine 1279 to glutamine, and its role in neurodegeneration and AD. Then I'll briefly go into some future directions and therapeutic implications of our work. Now, this was a study that was led by a very talented research assistant professor in my group, Leah Cuddy. And Leah did really an incredible tour de force characterization of a knock-in mouse that was made with this rare variant that is associated with Alzheimer's disease in humans. So this study could not have been done without Leah's hard work and dedication. So she gets all the kudos, all the credit for this. I'm just merely sitting around enjoying the show. But thank you, Leah, for this great work. It was just published in Science Translational Medicine on September 30th, and we were honored by the journal choosing Leah's study for the cover of the September 30th Science Translational Medicine. So just to give you, I know everyone's familiar with Alzheimer's disease, but I just want to put everything into context here. A short primer on Alzheimer's disease. Everyone knows about the severe brain atrophy of the disease, the neurodegeneration, the hallmark amyloid plaques and neurofibrillary tangles, the gliosis and neuroinflammation. I just want to point out three unsolved mysteries of Alzheimer's disease that I think that our work may provide some insight into, such as age. The first is age as a primary risk factor. Why does the incidence of Alzheimer's go up with age? Why do certain neurons of the brain become vulnerable to Alzheimer's disease degeneration, whereas others are resistant? And also, do women have a higher risk of Alzheimer's than men, even after you correct for longevity? These are mysteries that are still enigmatic today. And since we're going to be looking at a lot of pathology sections today, I want to just introduce the cellular players of Alzheimer's disease, which you all know about. First of all, the neuron and, of course, the lesion that forms in the neuron, the neurofibrillary tangle, which is composed of hyperphosphorylated tau. And then there are the innate immune cells of the brain, the microglia, that respond to the amyloid plaque, which is composed of the A beta peptide, and then the astrocytes, which are also responding to the amyloid plaque. And they're also involved in the immune response, the inflammatory response of the brain. This is the genetic landscape of Alzheimer's disease shown here. And over the last 20, 25 years, a lot has been discovered about the genes that are involved, that are associated with elevated risk of Alzheimer's disease. This is a plot here with, on the y-axis, you have low risk, medium risk, and high risk. And then on the x-axis, the frequency of the gene mutation, whether it's rare or common. Most notably, there have been the autosomal dominant mutations in the 3 genes for amyloid precursor protein and presenilin 1 and presenilin 2. These cause early onset aggressive forms of Alzheimer's disease that affect the production, that increase the production of the A beta 42 peptide. Now, a number of genome-wide association studies, and more recently, whole exome sequencing and whole genome sequencing studies have informed about the biology of Alzheimer's disease. And these 80 genes have been valuable for understanding pathways that are central to disease onset and progression. Most importantly, those variants that are in the protein coding regions of genes provide the most valuable biological insight. Many of these AD risk genes affect the metabolism of A beta, but most notably, more recently, a number of these, perhaps almost half, are involved in the innate immune system and are expressed in microglial cells, the innate immune cells of the brain. That's an interesting sidebar, which I think plays into our story a bit. And this is called a Manhattan plot. It is basically, the blue bars are all the risk factor genes, and the height of the bar tells you how strong they are in terms of their effects on Alzheimer's disease risk. Now, angiotensin converting enzyme, or ACE1, has recently become a validated AD risk gene. By that, I mean that it's above the threshold of validation that the geneticists have determined by their will to be 5 × 10⁻⁸ p-value. And here's ACE1 circled here. So it's just above the threshold. These are papers that came out after we embarked on our study in 2018 and 2019. But when these papers came out, they really gave us a shot in the arm, telling us that we were on the right track with this work, that ACE is really important, an important risk factor for Alzheimer's disease. Now, this is angiotensin converting enzyme shown here diagrammatically. It is a type 1 membrane zinc-dependent dipeptidase with 2 homologous extracellular catalytic domains, a C domain and an N domain here. Now, both of these domains have catalytic function. They are proteases, and they have a set of substrates that they cut, like molecular scissors. Angiotensin 1 being the most important for blood pressure control. but there are others. Bradykinin also controls blood pressure. Interestingly, A beta 42 is a substrate of ACE, and in vitro at least, ACE cleaves A beta 42. So one would think that could be protective. ACE is expressed in significant levels in all human tissues, including neurons in the CNS. But the level of ACE that's expressed is different depending on the neuronal subtype. For example, there are high levels of ACE in cerebellar neurons, but low levels in hippocampal neurons. We don't yet understand why that is. So we think it may be important in terms of ACE function of the brain, and we're very interested in that particular question. Another characteristic of ACE is that it's membrane bound. You can see right here, there's a little tail. This is the membrane here. There's a transmembrane, transmembrane region that anchors ACE into the membrane. There's this very short cytosolic tail, which I'll tell you is important in a minute. But what I want to point out here is that there's another protease called an ACE shed ACE that comes along and cuts right here to release this catalytic domain into the circulation, where it can go and cleave angiotensin 1 into 2, angiotensin 2, and thereby control blood pressure. Now, ACE is a component of a much larger system called the renin-angiotensin system, or RAS. This is the pathway here where the precursor protein, angiotensinogen, is cut by the protease renin into angiotensin 1, which is in turn cut by ACE into angiotensin 2. Angiotensin 2 then binds to the AT1 angiotensin receptor to perform the various functions of ACE in all these different tissues, including the brain. And angiotensin receptor activation in the blood vessels is responsible for constriction of the vessels and increasing blood pressure. So the functions of the RAS are very complex, involve many tissues of the body. What's not shown here, but I think it may be important, is that ACE is also has a role in the peripheral immune response by modulating the function of neutrophils and macrophages. ACE is highly expressed in monocytes upon differentiation into macrophages and dendritic cells, but it's also expressed at higher levels in microglial cells of the brain when they become activated. Those are the innate immune system cells of the brain, and they are also of a monocyte lineage. I think that may be important for our work. We still are working that out. But there have been some past clues as to the role of ACE1 in Alzheimer's disease. First of all, midlife hypertension has been associated with increased AD risk. And most recently, there's been the SPRINT Mind study that was published last year, which reported that people on antihypertensive drugs, irrespective of the class, at least they didn't really pay attention to the class of drug, but that those individuals on antihypertensives had a lower risk of developing mild cognitive impairment, and that there was a trend toward reduced dementia risk, although it wasn't significant. But as I said, the study did not differentiate between the different classes of drug. So we don't really know in that study if there were particular classes of drugs that performed better at reducing the risk of AD. Epidemiological studies suggest that the ACE inhibitors and the angiotensin receptor blockers for hypertension also reduce the risk of AD, and the ARBs are more effective than the ACE-Is, but we don't really understand why. There's also a common insertion of an ALU sequence in intron 16 of the ACE gene, which is associated with increased AD risk, and also it's associated with reduced hippocampal volume in women and decreased ACE1 serum levels. But the mechanism of how this ALU sequence insertion is affecting ACE levels is unknown, or how the ACE1 level may be increasing the risk of AD. That's still, that study did not address that question. It's been known that ACE1 levels are increased in postmortem AD brains. A number of groups have shown that ACE1 degrades A-beta in vitro, but A-beta degrading effects of ACE1 in vivo have been inconsistent. Some studies have shown that ACE1 elevation is reducing amyloid levels, whereas others show that there is no effect, or maybe even an increased effect. So it's been very inconsistent in terms of these studies. I've already mentioned that ACE1 is a validated AD risk factor gene by GWAS. So this is where we get involved, and our collaborator Rudy Tanzi, who is at Mass General Harvard, is a geneticist, and for a long time has been interested in genes that increase the risk of Alzheimer's disease. In fact, he was one of the first to discover the amyloid precursor protein gene. And he was been performing whole genome sequencing of families to identify rare AD-associated variants in protein coding regions of genes, which of course are the most biologically informative. And in one analysis of 446 AD families, he showed that he found that there were several rare coding variants in the ACE1 gene. One of these rare variants, ACE1 arginine 1279 glutamine, was present in 10 families with 13 affected carriers, 2 carriers with a mother-child relationship, and only 2 unaffected carriers. Now, the age of onset of this particular variant is about 70 years of old, 70 years age of onset, which is relatively young for late, this still classifies as late onset Alzheimer's disease, but it's a relatively earlier onset form of late onset, if you follow me. Rudy also and his team also found additional AD-associated rare coding variants, these here, which we're currently investigating. The ACE1 R1279Q was chosen for further analysis because of the strong association with AD families. Knock-in mice were made generated by the CRISPR-Cas9 genome editing mechanism system to be able to express the cognate variant in the mouse, which is ACE1284Q. So ACE R1284Q, the numbering in the mouse is a little bit larger because there's a 5 amino acid insertion in mouse ACE, but it's at the this mutation is at the very same position as that found in the humans that is associated with AD. So these mice were made on a Black 6 background. This table here shows a bit of the demographics of the cohorts for the whole genome sequencing and exome sequencing that Rudy used to identify the genes. This is when we got involved, and Leah got the mice to begin her characterization of the phenotype of these ACE1 knock-in mice, which I'll either refer to them as the variants, the knock-in mice, or the variant, rather than to keep repeating that number. I want to tell you a little bit about the variant and what it may be doing at the C-terminus of the transmembrane domain. So here is the very C-terminal portion of human and mouse ACE1. They're highly homologous. This is the position of the arginine that is changed to glutamine. The blue is the transmembrane domain. The green is the C-terminal tail. And most type 1 transmembrane proteins that we're familiar with, like amyloid precursor protein, have a string of positively charged glutamines or, arginines or lysines that tend to anchor. They perform a stop transfer function during translation into the ER, and they determine the topology of the transmembrane domain and anchor the protein in the membrane. As you can see here, ACE it's quite a large protein, as you saw from the previous diagrams, but instead of having a string of arginines, there's only one. I mean, there's another arginine over here, but that's only 2 positive charges in the tail. It doesn't seem like it's a lot of positive charge to be able to orient this membrane. And when this positively charged arginine gets replaced by the small uncharged glutamine, our hypothesis is that the reduction of this positive charge at the end of the transmembrane domain of ACE may affect the properties of ACE in the membrane, such as membrane stability, orientation of the membrane, and the membrane shedding by the shed ACE that releases the catalytic domains into the circulation. So this is the diagram again of ACE anchored to the membrane. Here is where the mutation is located by the red star. This is in a very unusual, this type of variant is very unusual to affect the properties of an enzyme. Usually you think of amino acid variants in the catalytic region of an enzyme would be changing the function of that enzyme. So this is an odd position. It's like the tail wagging the dog. And we really think it might have something to do with the properties of shedding, because one of the first experiments that Leah did was to isolate primary forebrain neurons from embryos of knock-in mice and culture them in vitro, and then look at where ACE was. She did cell surface labeling for ACE and found that the knock-in neurons had higher levels, about 1.5-fold more ACE1 on the cell surface of the knock-in neurons compared to wild-type neurons. She then measured the level of ACE in the cell lysate, saw a similar increase in ACE in the knock-in lysates. And interestingly, the shed ACE that was cleaved and released into the media was reduced. So again, it's supporting this idea that full-length ACE, membrane-bound ACE, is accumulating on the cell surface and is not being shed properly by the shed ACE. Leah also found that the primary neurons in culture, just when you let them culture by themselves without any stresses or treatments at all, they undergo spontaneous apoptosis, as shown here, down here with tunnel staining shown in red. There was dramatically increased tunnel staining at 7 and 14 days. Tunnels is a marker of apoptosis, as is cleaved caspase 3, which is shown here in the knock-in by Western blot and measured here, a massive increase in cleaved caspase. Also, markers of apoptosis, phosphorylated histone, H2AX is also elevated. So these neurons are just kind of set to be able to die, right? They've got a very hair trigger on apoptosis. Because midlife hypertension is associated with increased risk for AD, Leah wanted to know what was the blood pressure like in these knock-in animals. So we aged cohorts of these mice, and then Leah looked at blood pressure at 8 months and at 14 months and found no difference in either systolic or diastolic pressure. So blood pressure was normal. We then collaborated with Daniel Processy over in the small animal MRI core, and he found by MRI that there was no difference in cerebral blood flow or blood-brain barrier permeability. So the variant does not appear to cause significant changes in blood pressure or other cardiovascular or cerebrovascular functions. So it's likely that the effects of the variant are directly on the CNS. In other words, a mechanism that's occurring in the brain itself rather than in the periphery. Leah then looked at ACE1 levels in the brains of these mice. This is a knock-in brain, sorry. And you notice the green is ACE, and you notice there's higher levels of green ACE in the knock-in brain. These are cortical neurons. You can see that ACE1's labeling is elevated using this ACE1 antibody in the knock-in compared to wild type. These are cerebellar neurons. The cerebellar neurons of the knock-in have higher ACE. Also, co-labeling with neuronal marker NUN shows high colocalization, but there's little colocalization with the microglial marker IBA1 or the astrocyte marker GFAP. So we conclude that ACE1 is predominantly expressed in neurons rather than microglia or astrocytes. Leah then did Western blotting of different brain regions, cortex, hippocampus, and cerebellum in wild type and knock-in mice. Different regions of the brain express different levels of ACE. And you can see right away that the knock-ins had higher levels of ACE irrespective of the brain region. Now, there was this interesting inverse proportionality between ACE and renin and the angiotensin receptor. So brain regions that had high levels of ACE, like in the cerebellum, those regions had lower renin and angiotensin receptor, and the opposite was true for regions that had low levels of ACE, like the cortex and the hippocampus, which had higher levels of renin and angiotensin receptor. Of course, the knock-ins had higher levels of ACE. It even seemed to us that the receptor was depressed even further in the cerebellum with that more elevated level of ACE. So there must be, we suspect at least, that there's a homeostasis playing in the brain where high levels of ACE are compensated for by lower levels of receptors so that the signaling mechanism doesn't get out of whack. Aging also increased ACE, and shown here are homogenates from 3-month and 12-month-old wild type and knock-ins. This is showing that in the wild type brains, there is a normal, natural increase in ACE1 level from 3 to 12 months. So aging normally increases ACE1 levels. We don't understand why. For the knock-in, the knock-ins are already starting at a higher level of ACE at 3 months, and this level is increased even further at 12 months. There was also an increase in the knock-in brain in angiotensin 2, which is the product of cleavage by ACE1 and is the protein that activates the angiotensin receptor. The angiotensin receptor signaling is through the ERK pathway, and in the knock-in mice, we see elevated phosphorylated ERK as shown here. So aging is normally increases ACE. The variant increases ACE further. Just as a sidebar, angiotensin 2 has been associated with neurodegeneration in Parkinson's models. So perhaps this increase in ACE and angiotensin 2 specifically may be a general phenomenon in neurodegeneration. Leah then looked at the brains of these aged knock-in mice and found an age-associated atrophy. At 8 months of age, we have wild type here, knock-ins, and heterozygous knock-ins and homozygous knock-ins. At 8 months of age, there was really no effect on atrophy, but at 14 months, Leah found that there was a gene dose-dependent, knock-in gene dose-dependent increase in atrophy, as you can see here with the massively increased ventricle. She also measured hippocampal volumes. There were really no change to speak of at 8 months of age, but at 12 months of age, there was a gene dose dependent decrease in hippocampal volume and dentate gyrus volume, and an increase in the ventricular volume. And this is indicative of an autosomal dominant effect for this particular variant. When Leah then zoomed in to look at neurodegeneration in the hippocampus, this is what she found. The hippocampus of the knock-in showed regions of CA1 and CA2 in particular that showed neuronal loss. Red is the neuronal marker nu-n. At higher magnification, you can see that there is neuron loss in this region. The arrows are pointing to pycnotic nuclei, which are indicative of apoptosis. And what was interesting, this is a selective neuronal loss specific for the hippocampus. Other regions of the brain, like cerebellum and cortex, do not show any reduction in neurons, but the hippocampus does. This is nu-n covered area shown here. So there's a selective loss of hippocampal neurons in the knock-in mice. Other brain regions appear more resistant to neurodegeneration. The knock-in mice have also increased neuroinflammation and decreased synaptic markers. So, and this is more severely affected in females. So as one would expect, if there's neurodegeneration, then the innate immune system cells of the brain, the microglia, and the astrocytes would become involved to help clear up the damage. And that is indeed what we see by Western blotting. The microglial marker IVA1 is elevated, as is the astrocyte marker GFAP. The presynaptic neuronal marker synaptophysin is significantly decreased, and that's observed here, especially in the hippocampus. You can see the reduced, these are all stained and imaged at the same, under the same conditions. You can see in the hippocampus reduced synaptophysin labeling in the knock-in compared to wild type. And it may be a little bit hard to see here, but these are combined male and female points here. The open triangles are the female knock-ins, and the closed triangles are the males. And you can see that females consistently have more synaptophysin loss and more elevated IBA1 and GFAP. So the females are more affected. Also, the females, but not the males, have behavioral hippocampus dependent memory deficits. Both in the Morris-Water maze, time to platform was increased. Platform crossings were abnormal, as was at least one of the quadrant times. Fear conditioning was quite dramatically affected in the cued fear conditioning as well as contextual. So the females, but again, not the males, I'm not showing you the male data, the females had a functional deficit. For some reason, we don't yet understand fully what the basis of this sex difference is, but it harkens to the notion, at least is reminiscent, that women have a higher incidence, higher risk of Alzheimer's disease than men. We also did EEG in the hippocampus and the prefrontal cortex. And again, the females, but not the males, had abnormal EEGs. These are different rhythms, and the power spectral densities are abnormal in certain rhythms during the day and many rhythms at night in the female mice. But the prefrontal, and this is in the hippocampus, but the prefrontal cortex doesn't have very much difference. That's illustrated here with the dominant frequency daily averages. Hippocampus is elevated in the female knock-ins, but no change in the prefrontal cortex. This is during the day and also at night. So there are functional differences and electrophysiological differences in the females. Although the neurodegeneration in both sexes is about the same. One important point is that inflammation is more strongly affected in the females. So perhaps females are more susceptible to neuroinflammation than males are, and that may be the reason why there are these more dramatic functional differences. Now, one of the most important experiments that Leah did was a rescue experiment with drugs, antihypertensive drugs that block either ACE1, the ACE1 receptors, or the ACE1 inhibitors, or the receptors of ACE, the angiotensin receptor blockers. She used two drugs, captopril, which is an ACE inhibitor, and losartan, which is an angiotensin receptor blocker, and found with a 6-month treatment in water, so this was ad lib in the drinking water, she found that, so here's the vehicle, you can see that the hippocampus volume is dramatically reduced in the knock-in mice that are vehicle treated compared to wild type. But this is completely rescued when you treat these mice for 6 months, prevented, I should say, with either the ACE inhibitor or the ARB. This is also true for the decrease in dentate gyrus volume, and also the new uncovered area is also rescued. It looks like the ARB is doing a little bit better than the ACE inhibitor in terms of the rescue. And for example, well, you can't really see it here, but I just want to point out this is the CA2 region of the knock-in, which has severe neurodegeneration, which is completely rescued by captopril and losartan. And both of these drugs do cross the blood-brain barrier, but there are other drugs in their classes that do have a better job of crossing the blood-brain barrier that we're interested in trying. We did measure the level of these drugs, the concentrations of these drugs in the brain. Captopril and losartan got equally well into the brain of the knock-in compared to wild type. No differences there. There was quite a spread in the amount of losartan that got into the brain, high variability there. But even at the very lowest concentration of losartan, the ARB did produce a rescue effect. And so another indication that the ARBs may be more effective at preventing neurodegeneration. Captopril, on the other hand, had a positive correlation with hippocampal volume, shown here. But at the lower concentrations of captopril in the brain, there was still significant neurodegeneration. So, but this was an important experiment to validate that the effects of the neurodegeneration are mediated through elevated ACE1 activity. And then when you block that ACE1 activity, either through an ACE1 inhibitor or an ARB, you can rescue the neurodegeneration. You can prevent the neurodegeneration. You can also prevent the inflammation, the neuroinflammation, and this is GFAP in captopril and losartan treated wild type or knock-in mice. Here's the quantification here. This is IBA1 for microglial cells. Again, you can see that the elevated astrogliosis and microgliosis are returned to normal. So one of the final experiments that we did was to cross the knock-in mice with our amyloid 5XFAD mice to see if there's an interaction between ACE1 and amyloid. So the 5XFADs on an ACE1 wild type are shown here. Green is amyloid, red is nu-N for neurons. You can see that at 6 months of age now, that the amyloid from the 5X mouse has really decreased the age of onset of neurodegeneration. Normally, in the knock-in itself, neurodegeneration starts at about 12 to 14 months. So amyloid, in the presence of amyloid, neurodegeneration was significantly accelerated. And there was a gene dose dependent effect where the heterozygous knock-ins and the homozygous knock-ins had the greatest volume decrease compared to the heterozygous and wild type. So this was really an important experiment to show an interaction, but and I might add that there was also a increase in the microgliosis and astrogliosis as shown by IBA1 and GFAP, as one would expect if there was more severe neurodegeneration going on. But interestingly, the levels of amyloid were unchanged. And so what this experiment shows is that although the knock-in does accelerate the neurodegeneration, the knock-in itself, the variant does not affect A beta levels themselves. And this kind of, in my view, is pointing toward that ACE1 does not appear to be an A beta degrading enzyme in the brain under normal conditions. Perhaps in vitro it is, but not in vivo. So this is a rare coding variant, only occurring in a few families that we've identified so far. Does this have anything to do with common garden variety Alzheimer's disease? Well, as I told you earlier, ACE1 levels are increased in Alzheimer's disease brain, and we replicated that here. It's about a twofold increase in the level of ACE1. There's a trend for the receptor to be decreased in the AD brain. But this is really dramatic in the controls. So the controls are black. You can see that there's a strong negative correlation between angiotensin receptor and ACE1 level. So in the normal brain, there's a homeostasis going on where if you have high levels of ACE, you have low levels of receptor to balance, I believe, to balance the signaling that's going on in the brain. This homeostasis, this negative correlation is lost in AD, where the correlation is flat. We also did staining for ACE1 in human AD brain sections, and we see good colocalization in both the control neurons with ACE1 staining as well as in AD, although there are fewer neurons here in the AD brain. This was quite an advanced AD stage where there was quite a bit of neurodegeneration going on. But we think that because of this elevated ACE that is common in Alzheimer's disease, that perhaps overactivation of this pathway may be contributing to neurodegeneration in normal garden variety AD. So this is our working model. The variant increases neuronal ACE on the cell surface. And this, of course, higher levels mean higher activity. More angiotensin is produced, angiotensin 2. This leads to hippocampal neurodegeneration, either through a cell autonomous or non-autonomous mechanism where, in the case of hippocampal neurons, they express low levels of ACE, but they have high levels of receptors. So they may end up committing suicide by producing angiotensin 2 and activating these apoptosis pathways. But there's also a non-cell autonomous pathway in which some cells that have a high level of ACE, like the brainstem or cerebellar neurons, make a lot of angiotensin 2. They themselves have low levels of receptor, so they don't kill themselves. But maybe they synapse upon other cells that have high levels of receptor, like the hippocampal neurons, and they end up killing. If ACE is overactive, they end up causing neurodegeneration in those downstream neurons. So I think in the end, this knock-in may provide insights into the mechanisms of aging in AD, selective neuronal vulnerability of certain neuronal types, why some are vulnerable and why some are resistant, and why females are more susceptible to Alzheimer's disease. There's also, borrowing from the cancer field, a second hit that may be occurring with not only aging, which is also increasing ACE1 levels normally on its own, but also the Alzheimer pathologies, amyloid and tau, could be accelerating the neurodegenerative process in the conditions where you have higher levels of ACE. And so I think it's really important now for us to consider doing prevention trials with better blood-brain barrier penetrant ACE inhibitors and angiotensin receptor blockers. And that's for the future. I'll skip over the summary, which is very wordy. Future directions, Leah is going to be doing single nucleus RNA sequencing of hippocampal cells from the knock-ins to get a better handle on the transcriptomic processes that are occurring in individual cells as those mice age. She's also characterizing forebrain neuron conditional ACE1 knockout mice, because we really don't understand the normal function of ACE1 in neurons. That's a fundamental question. She's also done an experiment with telmisartan, which is an ARB that has probably the best blood-brain barrier penetration. And we want to see how well that protects these knock-in mice. We're also interested in these other variants that the Tanzi group have identified and what their mechanisms might be. Are they similar to the mechanism? Is there a common mechanism that ACE1 has in Alzheimer's disease? And we're also planning this R01 for the February deadline coming up. So with that, I want to thank everybody on the ACE1 study, Leah and her team, also the collaborators, Rudy and his group, as well as Daniel Processi in the small animal MRI core. And I really want to thank the Vassar Lab, which here they are from left to right. This is Leah right here. I want to thank them for their great dedication, their hard work and enthusiasm. I also have a number of acknowledgements down here. And of course, I want to thank the Cure Alzheimer's Fund, which funded all of this work. And there still are a couple minutes for questions. So I think I'll end there and I'll stop sharing my screen. Thank you very much.
Okay, thanks, Bob, for a really stimulating look at a new area of Alzheimer's disease. Lisa, I am not seeing question. Okay, let's see, question and answer.
It looks like right now no one submitted questions, so feel free to do that in the Q&A chat box, or you can, there's a feature to raise your hand and I can let you ask a question.
Okay, so if there are no, while we're waiting, number one, there is 4 or 5 minutes in the hour itself. Lisa, can we extend questions for 5 or 10 minutes perhaps, or how does that, will that work? Bob, are you willing to do that if we have? Okay.
So
good.
Do we have time for questions?
We do.
Yes.
Great. Okay, so
Marcel,
Bob,
do you have a question?
yeah, and comment. So Bob, beautiful talk and also beautiful pictures. I'm not surprised at all that it made the cover. Just a couple of comments. So you know the ACE inhibitors have been around for a very long time, especially losartan and similar inhibitors. And of course, you know, the existing literature of whether this helps or doesn't help Alzheimer's disease and so on, very complicated. And it's not surprising because there's a lot of noise in the system. But I wonder if it may be worth doing things a little differently. For example, now throughout the Alzheimer's centers, there's a huge amount of CSF that's available, including metabolic imaging. So I think the question to ask whether CSF inflammatory markers, for example, might be different in people taking ACE inhibitors that cross the blood-brain barrier may be something worth keeping in mind. And then I have just a couple of questions that obviously, you know, SARS coronavirus 2 gets into the human cells through ACE2 inhibitors. So what is the relationship of ACE2 to ACE1? And finally, my question is, what happened to the blood pressure of your mice?
Oh, well, you know, I'll answer that first. I showed a little bit earlier on in the talk that one of the first experiments Leah did was to measure blood pressure in those knock-in mice, and there was no difference. The knock-in did not cause any change whatsoever, either in systolic or diastolic blood pressure, nor in any of the cerebral vascular properties, functions, blood flow. Cerebral blood flow was unchanged, as was blood-brain barrier penetrance or permeability. So there wasn't really an effect on any of the what you would normally think ACE would be doing peripherally. Probably that's because there's a homeostasis going on with ACE and with angiotensin in the periphery. There have been a number of mutations that one would that people have studied regarding their effects in various conditions, and you think that these mutations in ACE would affect blood pressure, but they don't. So the RAS system is very good at homeostasis and adjusting other parts of the system. And then what was your other question? I'm sorry, I forgot the.
So the relationship of ACE1 to ACE2.
Oh, ACE1 to ACE2, they're highly homologous. There's no evidence that I know of that CoV2 binds to ACE1. The inhibitors, on the other hand, may be upregulating. You know, the inhibitors of ACE1 may cross and inhibit ACE2. Now that cross inhibition may cause an upregulation of ACE2. Now that could be a risk factor for COVID. That's what I've heard, but I don't know a lot about that. I don't think that ACE2 is playing a big role. At least we have to look into that. That's another question about ACE2 role in the brain. ACE1 and angiotensin receptor 1 seem to be the major players in the brain. But I'll check into that. That's a very good. And I liked your previous comment about looking at CSF. We could look also see correlations with inhibitors and markers of neurodegeneration like NFL. And one may suspect that NFL would be lower in the people taking the ACEIs and the ARBs, but that penetrate the brain. So that would be a good experiment to do.
And you know, you could also measure the level of ACE inhibitor that's in CSF, which would give you a measure of how it penetrates. So you could ask pretty specific questions.
Yeah, those are really great experiments. Thank you.
So Bob, I was wondering, toward the end, you indicated that the knock-ins don't affect amyloid level, but rather they affect neurodegeneration. So is that giving us any insight as to whether clinical trials, human clinical trials that are totally eliminating excess amyloid might be ineffective? Or is this something very specific to this ACE1 situation?
Well, I mean, that's a good question, John. I it is a possibility. It is a possibility that people with higher levels of ACE would be more susceptible to the effects of the lesions. And this may be one explanation, one possible explanation for why some people grow old perfectly cognitively normal with a head full of amyloid and they don't have Alzheimer's disease, but maybe they have low levels of ACE. And the angiotensin pathway is low in their brain, so they're not as susceptible to neurodegeneration. However, in some unlucky individuals that have higher levels of ACE and higher levels of angiotensin pathway signaling, if they start getting even a sniff of amyloid, their neurons may be starting to fall off the cliff. So that's an interesting hypothesis. And we just don't understand vulnerability and resistance and resilience. It may be one mechanism that we could achieve understanding of those conundrums right now.
Are there other questions? Lisa, I'm not.
I don't think I see any Q&A questions, but if anyone wants to hop on and ask a question, I can bring you into the webinar. Otherwise, we are about 3 minutes over, so.
Yeah. Well, if not, Bob, I want to congratulate you and your lab on a really impressive study that's going in a very new direction for Alzheimer's disease, and a wonderful, very informative seminar. And as Marcel said, beautiful pictures. Thanks.
Thank you all for your attention, and we'll be seeing you later.
Okay.
Thank you.
Thank you.
Thanks, everyone.
Bye-bye.